Welcome, curious minds, to a world of knowledge and discovery. Welcome, welcome, welcome. We're thrilled to have you here as we tackle Streptomyces griseus. Streptomyces griseus is a species of bacteria in the genus Streptomyces commonly found in soil. A few strains have been also reported from deep-sea sediments. It is a gram-positive bacterium with high GC content. Along with most other Streptomycetes, South griseus strains are well-known producers of antibiotics and other such commercially significant secondary metabolites. These strains are known to be producers of 32 different structural types of bioactive compounds. Streptomycin, the first antibiotic ever reported from a bacterium, comes from strains of South griseus. Recently, the whole genome sequence of one of its strains had been completed. The taxonomic history of South griseus and its phylogenetically related strains has been turbulent. South griseus was first described in 1914 by Krainsky, who called the species Actinomyces griseus. The name was changed in 1948 by Waksman and Henrici to Streptomyces griseus. The interest in these strains stems from their ability to produce streptomycin, a compound which demonstrated significant bactericidal activity against organisms such as Yersinia pestis, the causative agent of plague, and Mycobacterium tuberculosis, the causative agent of tuberculosis. Streptomycin was discovered in the laboratory of Selman Waksman, although his student Albert Schatz probably did most of the work on these strains of bacteria and the antibiotic they produce. In this chapter, we'll be unraveling the enigma of taxonomy and discovering its transformative power. Streptomyces is the largest genus of the Actinomyces and is the type genus of the family Streptomycetaceae. These are gram-positive bacteria with high GC content and are characterized by a complex secondary metabolism. They produce over 2/3 of the clinically useful antibiotics of natural origin. Streptomycetes are found predominantly in soil and in decaying vegetation, and most produce spores. Streptomycetes are noted for their distinct earthy odor, which results from production of a volatile metabolite, geosmin. Like other Streptomycetes, South griseus has a high GC content in its genome, with an average of 72.2%. The species was first classified within the genus Streptomyces by Waksman and Henrici in 1948. The taxonomy of South griseus and its evolutionarily related strains have been a considerable source of confusion for microbial systematists. Gene sequence data have been used to recognize the related strains and are called South griseus gene clade. The strains of this clade have homogeneous phenotypic properties but show substantial genotypic heterogeneity based on genomic data. Several attempts are still made to solve this issue using techniques such as homology and multilocus sequence typing. A whole genome sequence was carried out on the IFO 13350 strain of South griseus. Moving forward, we'll be taking a closer look at physiology and morphology. South griseus and its related strains have recently been shown to be alkalophilic, i.e., they grow best at alkaline values. Although these organisms grow in a wide range from 5 to 11, they show a growth optimum at 9. They produce grey spore masses and grey-yellow reverse pigments when they grow as colonies. The spores have smooth surfaces and are arranged as straight chains. In the next segment, we'll be exploring ecology and its implications for our subject matter. South griseus strains have been isolated from various ecologies, including still waste tips, rises for deep-sea sediments, and coastal beach and dune sand systems. Recent studies have indicated the strains of South griseus might be undergoing ecology-specific evolution, giving rise to genetic variation with the specific ecology, termed ecovars. Get ready for a thought-provoking discussion as we delve into antibiotic production and its impact on our understanding. Interest in the genus Streptomyces for antibiotics came after the discovery of the antibiotic streptomycin in a South griseus strain in 1943. The discovery of streptomycin, an anti-tuberculosis antibiotic, and Selman Waksman the Nobel Prize in 1952. The award was not without controversy, since it excluded the nomination of Albert Schatz, who is now recognized as one of the major co-inventors of streptomycin. The strains of this species are now known to be rich sources of antibiotics and to produce 32 different structural types of commercially significant secondary metabolites. Furthermore, the genomic studies have revealed a single strain of South griseus IFO 13350 has the capacity to produce 34 different secondary metabolites. As we venture forward, let's take a closer look at etymology and its impact on our understanding. By 1943, Albert Schatz, a student working in Selman Waksman's laboratory, had isolated streptomycin from Streptomyces griseus from the Greek strepto twisted plus mix and the Latin griseus, grey. In the upcoming section, we'll be dissecting the official New Jersey state microbe and exploring its implications in greater detail. South griseus was designated the official New Jersey state microbe in legislation submitted by Senator Sam Thompson R12 in May 2017 and Assemblywoman Annette Quiono D20 in June 2017. The organism was chosen because it is a New Jersey native that made unique contributions to healthcare and scientific research worldwide. A strain of South griseus that produced the antibiotic streptomycin was discovered in New Jersey in heavily manured field soil from the New Jersey Agricultural Experimental Station by Albert Schatz in 1943. Streptomycin is noteworthy because it is the first significant antibiotic discovered after penicillin, the first systemic antibiotic discovered in America, the first antibiotic active against tuberculosis, and the first-line treatment for plague. Moreover, New Jersey was the home of Selman Waksman, who was awarded the Nobel Prize in Physiology or Medicine for his systematic studies of antibiotic production by South griseus and other soil microbes. The bill was signed into law by NJ Governor Phil Murphy May 2019. Remember to follow me on social media for behind-the-scenes content and updates.